Hello, today we talk about how eukaryotic cells synthesize protein. Proteins are large and complex molecules made by chaining together amino acids. There are 4 structural levels of protein: primary, secondary, tertiary, and quaternary. This will not be discussed in detail right now. However, know that protein shape is critical to its activity. Proteins have a key role in linking the genotype to the phenotype. Moreover, they have many crucial functions. For example, human hair, antibodies, enzymes, and some hormones are all made of protein. The process of turning DNA into protein consists of 3 stages, which we will discuss in detail now.
Moving on to transcription, DNA double-stranded helix would be converted to a single-stranded mRNA, which consists of instructions for protein synthesis. This is the main enzyme RNA polymerase which is involved in transcription. So transcription takes place in 3 steps: initiation, elongation, and termination. Initiation takes place when RNA polymerase recognizes binding sites to the promoter on the DNA, and afterwards it binds to this site, and it requires help of additional transcription factors in the process. Afterwards, which unwinding of the DNA takes place by breaking hydrogen bonds between complementary bases, and this is perfectly shown in the analogy when opening a zip, as shown here. Afterwards, elongation takes place when RNA polymerase adds complementary RNA nucleotides to that on the DNA template strand, and complementary base pairing occurs by formation of hydrogen bonds, and the mRNA strand is synthesized from the 5' to the 3' direction, as you can see over here. And afterwards, phosphodiester bonds form between the added nucleotides, and that forms the new mRNA strand.
In termination, DNA sequences called terminators signal completion of the transcript. The transcript is released from the RNA polymerase. Sickle cell anemia is an example where transcription of a mutated DNA template strand results in a genetic illness. The next step of transcription is called RNA modification. The transcript must go through extra processing before it can direct translation. End modifications involve addition of a 5' cap and poly A tail. Splicing is when introns are chopped out and exons are stuck back together. RNA modification can be likened to movie editing. The primary transcript can be imagined as raw footage. The 5' cap is a title screen, and the poly A tail is the credits screen. Deleted scenes or introns are removed, and the remaining footage is combined to create a coherent story. Failure to regulate splicing has been linked to many types of cancers. This may involve incorrect removal of introns and attachment of exons.
The final step is translation. Just like transcription, translation also undergoes initiation, elongation, and termination. But let's first discuss translation machinery. Firstly, there's tRNA, which brings specific amino acids to the correct ribosome. Then there's also rRNA, forming the ribosome structure, and within the ribosome there's 3 main regions. The A site, where the tRNA first arrives with this amino acid, the P site, where bonds between amino acids are formed, and finally the E site, where tRNA, after dropping off its amino acid, exits the ribosome. mRNA is written in groups of 3 nucleotides, called codons. Anticodons on tRNA pair with these codons to deliver the correct amino acid to the right ribosome. An example of this is the start codon AUG, which codes for methionine. Now that we've covered machinery, let's start with step 1, initiation. Initiation starts when the small ribosomal subunit binds to the 5' cap of the mRNA and locates the start codon. Next, a charged tRNA carrying methionine then attaches to the P site of the small ribosomal subunit. Finally, the large ribosomal subunit joins, forming the initiation complex and preparing for protein synthesis.
Elongation is the second stage of translation. It involves an amino acid carrying tRNA entering the A site of the ribosome, where it complementarily base pairs the tRNA's anticodon to the mRNA codon. Once this occurs, the ribosome, the large ribosomal subunit, will form a peptide bond between the old polypeptide chain attached to the old tRNA to the new amino acid. This will then cause the old tRNA to enter the E site and be discharged as it's done its role, and the new tRNA will then move from the A site to the P site and will now be holding the new polypeptide, waiting for the next tRNA to repeat the cycle. This cycle again is very similar to a factory line, where we have a car that has wheels being added, then has windows being added, then has electronics, and then the next car shows up. So it's a very similar process that continuously adds amino acids until the protein length is formed. Termination. This is the last stage of translation. It's very simple. A stop codon is read, which is the last codon of the mRNA, which causes a release factor to enter the A site. Once this release factor enters, it does 2 things. It separates the last tRNA molecule from the polypeptide and disassociates the ribosome as it's completed its role. Now, is the protein done forming? Yes, but it's not finished structurally. It can have different modifications added to it, like a lipid, a phosphate group, amino acids may be added or removed. And it may be combined with a new polypeptide chain as well, another separately formed one. For example, hemoglobin, because it's a more complex, it's a quaternary protein. So these modifications happen in the end, and again, they're very similar to a chess set. How a chess set will have, a pawn will function as a pawn, but it may look different between sets, because they may have a different style in mind.
Thank you for listening.